Hello everyone, welcome to our YouTube channel pharm. In this lecture, we will study the concepts of meiosis. Lecture will include the introduction to meiosis, stages of meiosis, introduction and phases of meiosis 1, introduction and phases of meiosis 2 and significance of meiosis. So, let's start. Meiosis. It is also known as reproductional or reductional cell division. In this, a single cell is divided 2 times to form 4 haploid daughter cells, as this diagram shows both meiosis 1 and meiosis 2. As we know that meiosis has 2 stages, that is meiosis 1 and meiosis 2. Meiosis 1 is further divided into several phases as prophase 1, which has 5 stages, that is leptotene, zygotene, pachytene, diplotene and diakinesis. And other phases are metaphase 1, anaphase 1, telophase 1 and cytokinesis. Next is meiosis 2, which is further divided into phases as prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. This diagram shows the sequential representation of phases of meiosis 1 and meiosis 2. Now we will understand each and every meiotic phase in detail. Meiosis 1, it is a type of cell division crucial to sexual reproduction. The pair of homologous chromosomes separates and diploid cells get reduced to haploid stage, that is 2n. It is differentiated into several phases as prophase 1, metaphase 1, anaphase 1 and telophase 1. This diagram shows the meiotic phases according to their movement and division of chromosomes. Now, we will understand about the phases of meiosis 1. So, first phase is prophase 1 and prophase 1 is further divided into several stages. Leptotene, it is visible under microscope and beginning of condensation of chromosomes takes place and compaction of chromosomes continues in leptotene. Second is zygotene, homologous chromosomes start pairing and the process is known as chromosomal synapsis. And followed by synaptonymal complex that is transiently formed during meiosis to promote homologous recombination between maternal and paternal chromosomes. Third is pachytene. In this, crossing over of non-sister chromatids of homologous chromosomes takes place. And chromosomes stay associated at the crossing over sites. Fourth stage is diplotene. In this, dissolution of synaptonymal complex and tendency of recombined homologous chromosomes of bivalents to separate from each other except crossing over sites. These are known as chiasmata. Last is diakinesis. In this, termination of X-shaped chiasmata takes place. Chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosomes for separation. Nucleolus disappears and breakdown of nuclear envelope occurs. Diakinesis stage is the transition stage to metaphase. This diagram shows the stages of prophase 1. In leptotene, replicated chromosomes. In zygotene, synapsis begins. Formation of bivalent and crossing over has occurred in pachytene. And in diplotene, synaptonymal complex dissociates, and in diakinesis, there is end of prophase 1. Second phase of meiosis 1 is metaphase 1. According to this phase, bivalent chromosomes align on the equatorial plate. Microtubules from opposite poles of spindle fibers attach to the pair of homologous chromosomes as the below figure shows. And the orientation of maternal and paternal chromosomes of each bivalent is random. Next phase is anaphase 1. Bivalent chromosomes separate and move to opposite poles. And sister chromatids do not separate, means attached to each other. This diagram shows the separation of daughter chromosomes and sister chromatids attached to the centromere. Last phase of meiosis 1 is telophase 1. Nuclear membrane and nucleolus reappear. This phase is followed by cytokinesis, and this gives rise to a dyad of cells as the below diagram shows. Now we will start with meiosis 2. It resembles to mitotic phase. Meiosis 2 is divided into phases such as prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Diagram shows the movement of chromosomes during meiosis 2. Next topic is phases of meiosis 2. So, first phase is prophase 2. It is initiated after cytokinesis when daughter cells are formed. And it begins the condensation of chromosomes. At the end, nuclear membrane, Golgi apparatus, and endoplasmic reticulum disappears. Formation of spindle fibers at each pole and compaction of chromosomes also begins. Diagram shows the reformation of spindle fibers and breakdown of nuclear membrane. Next phase is metaphase 2. In this phase, the sister chromatids are completely condensed or untangled. The chromosomes are connected to the centriole at the kinetochore of sister chromatids through the microtubules. And they also get aligned at the equator to form the metaphase plate. Here, this is the metaphase plate and chromosomes align at center, and here spindle fibers are attached to centromere. Third phase of meiosis 2 is anaphase 2. There is a simultaneous splitting of the centromere of each chromosome. Sister chromatids are pulled away towards the opposite poles, and at the same time kinetochore is at leading edge with chromosomal arms tailing. And non-kinetochore microtubules promote the elongation of cell. In this diagram, it shows the separation of daughter chromosomes. Next and last phase is telophase 2 and cytokinesis. According to this phase, chromosomes begin to uncoil or untangled and becomes thin. Reformation of nuclear membrane and nucleolus in each pole. This phase is followed by cytokinesis, which separates the two cells into 4 different haploid cells. Telophase 2 marks the end of meiosis 2, and as a result 4 haploid daughter are formed. For example, in humans, genetically 4 different haploid daughter cells are formed in meiosis, each with 23 chromosomes. As genetic differences ensure siblings of same parents are never entirely genetically identical. This is a figure of telophase and cytokinesis. Cytoplasm starts to divide and nuclear membrane reforms in telophase 2. Here, in cytokinesis, nucleolus reappears and 4 haploid cells are produced. Now, our last topic is significance of meiosis. Reproduction in animals takes place through the fusion of gametes, that is to cell fuses together with their genetic material to develop a zygote. It reduces the number of chromosomes in the gametes to half of their parent germ cells, that is to N. Meiosis restricts the multiplication of chromosome numbers and maintains the stability of species. Maternal and paternal genes get exchanged during crossing over. It results in the variations among the offspring. And all 4 chromatids segregates and go over separately to 4 different daughter cells. This leads to genetic variations in the daughter cells. In the next lecture, we will understand the concepts of cell junctions. Thank you. Keep watching and take care.